Hi. Good morning, everyone. My name is Dongkai Zhu. I'm a researcher from Office of Pharmaceutical Quality Research, OPQ Cedars. Today, I'm going to share with you some of results about how we develop in vitro release testing for complex drug product. Okay, so everyone deserves confidence in their next dose of medicine. So pharmaceutical qualities ensure the availability, safety, and efficacy of every dose. So this is the overview. So my presentation will have two sections. In the first section, I will talk about why we need better in vitro release testing methods for complex drug products, including challenges and needs. And in the second section, I will talk about our internal approach to solve these problems. In this section, I will use adaptive perfusion as examples, and then followed by a case study and some other potential uses. So in vitro release testing, also known as IVRT, has been designed for measure the in vitro release rate and extent of the drug release. And IVRT has been widely used for product development, quality control, and bioequivalence studies. So the pictures here are common IVRT methods. So the methods that with blue name tags are compendial methods, for example, USB apparatus 127, and the methods with red name tags are the non-compendial methods. So both of them can be used to measure the in vitro release behavior for complex drug product. So the scope of this talk is focused on the challenge of the developing IVRT method for complex drug product like ophthalmic emulsions, protein bonded complex, and also the long-acting injection product and nanomaterials. So you might wondering what a good IVRT method, what kind of IVRT method is a good considered as good. So a good IVRT method needs to be reproducible, which includes being precise and robust, and also it needs to be discriminatory, which including sensitivities and selectivity. So the goal of IVRT, like I just mentioned, is to measure the in vitro release. The goal of IVRT is to measure the in vitro rate and extent of the drug release by separating the free drug from its other carriers. And the common ways to use this is to membrane diffusion. So which is, and this method can be applied to many type of drug product like Dr. Robinson liposome injection and paclitaxel protein bonding injectable suspension, nano emulsions, and nano suspensions. So, however, the membrane diffusion does have the limitations because it's governed by the concentration gradient from the high concentration to the low concentration side. And also, the diffusion membrane is slow. So usually it takes about 2 to 3 hours for 90% of the drug come across from the membranes. So compared to the drug release, which usually take about within a couple of minutes. So the membrane diffusion method may become a rate limiting step and cannot capture the entire process. For example, the release process you saw may not be the truly release process. For example, some of the complex formulations that exhibit multi-phase release, like birth release at the earlier stage, the membrane diffusion method may not be captured these features. So now I would like to use the ophthalmic emulsions as examples to further discuss these questions. Ophthalmic emulsions, because it's considered as one of the type of the complex drug product, and it can be challenging because it may contain active ingredients like cyclophorin, and also ophthalmic emulsions have complex dosage form and may exhibit multi-phase release characteristic. And also ophthalmic emulsions, it has contained the complex route of delivery, which is through the eyes. So all those features are make ophthalmic emulsion is a good example for complex drug products. So here is the result that we did for the that we use reverse dialysis as the IVRT method for measure the in vitro release behavior for diphoprolate ophthalmic emulsions. The result on the red shows that in these studies we test like two different formulations with different globule size distributions. On the right side, you can see there's no discrimination was observed between this formulation in terms of the release profile. And also there's no multiphysics behavior was observed. And because of this too slow release rate, product degradation was detected. So this kind of highlights the limitations of the traditional dialysis-based IVRT method. So the challenge with the current IVRT method can be divided into two sections, which is reproducible and discriminatory. So from the previous example, we saw the reproducible is well presented in the result by seeing that's really tiny error bar in the figures. So how can we improve this IVRTs? So there might be different ways to do that, to tackle this question. And for example, like by developing any other type of in vitro release testing system. So here is an example that I'm going to talk about is that what we did in our lab. This IVRT method named as adapter perfusion system. So adapter perfusion system is utilizing the tangential free filtration principle to study the drug release. So compared with the normal filtration process, there will be the feed flow for this method will be parallel to the membrane filters. So there will be really less accumulations on the membrane surface. So anything that accumulates on the surface will be swiped out by the feed flows. Also, anything that's smaller than the pore size of the filter will be forced to migrate through the pore size and to achieve the separations. So there's two vessels are used in this method. One is named as retentive vessel. One is named as permeate vessel. So the retentive vessel is used to study how much drug was remaining in the system, and the permeate vessel is used to study how much drug was released in the system. Because the entire system was recording, so we are easy to calculate the rate and extent of the drug release. So this is a schematic of the adapter perfusion system. So if you see in the middle, the blue vessel is the retentive vessel, and where you inject samples here and mixing with the dissolution medias. And the gray vessel on the left is a fresh pump reservoir. It will continue to pump fresh media to the retentive vessel to maintain the constant volume in the retentive vessel, which is very critical to calculate the drug removal rate. And the green reservoir on the right is a permeate reservoir, which is to collect the drug release the release drug. And let me explain how this system works. So at the beginning, so you inject sample into the retentive reservoir, which is like the red line here, and the mixture, and then pump through the TFI filters and go back to the retentive reservoir, followed by the blue line. And the back pressure controlling valve here will create a transmembrane pressure that is perpendicular to the feed flow inside the TFI filters. And this pressure will serve as a driven force that forces those free drugs that's smaller than the pore size of the TFI filter to go through the permeate pathway and collect by the permeate vessels. And because of the continuous pumping from the fresh medium, the liquid volume in the retentive reservoir will be stay constant. That is really critical. So for us to calculate the drug release rate. And here is a case studies. So we are using adapter perfusion as an IVRT method to study the diphoprolate ophthalmic drug ophthalmic emulsions, and we intentionally create 3 formulations that with different globule size distributions. And in this study, we also include the pure drug solution and drug-loaded micelle solutions. And on the left figure, you can see the rate and extent of the drug has been removed from the retentive reservoir. And on the right figure, you can see the in vitro release behavior for these 5 formulations. So what we can see from the left figure is that really like 80% of drug has been released from the solution in less than 10 minutes. And similarly, what we saw from the red figures is that almost 90% of drug has been released to the retentive to the permeate vessel within 30 minutes. So both of the figure shows that adaptive perfusion can achieve the fast release, and the figure on the permeate side too shows that adaptive perfusion has a good discrimination capabilities for those formulations. And we also compared the adaptive perfusion with traditional dialysis methods with using the pure drug solution and 2 formulations that have small and large globule size distributions. So as you can see from the left, adaptive perfusion, you can see how fast adaptive perfusion shows drug release compared with the traditional dialysis. And this the red figure also provides more direct evidence, shows that adaptive perfusion is a fast and discriminatory IVRT method for ophthalmic emulsions, which is typically one type of the drug product. So now we are kind of confident that adaptive perfusion is a reproducible method, is an IVRT method that's reproducible and discriminatory. So after that, so we have one step forward, so where we implant the automation function to the adaptive perfusion method. So the figure on the left is our automated adaptive perfusion system. Where we design our own programs that can achieve automatically like precondition process experiment and watch the system when the experiment complete. And we also develop an algorithm that can automatically maintain a constant volume in the retentive reservoir, which is, like I said, very critical to calculate the drug removal rate during the experiment. So this automation function dramatically reduces the human burden and allows the result to be consistent even if the system was operated by multiple persons. So the figure on the right shows that this automated adaptive perfusion system can generate similar result, compared with the manual operations. So this shows this system the result generated by this system is also trustworthy. So in addition to ophthalmic emulsions, there's another category of complex drug product, which is formulations that can contain nanoparticles, like paclitaxel protein bonded injection suspensions. So the challenge is for this kind of formulation is that we are lacking the technologies that can sensitively detect the rapid release process of these formulations. And we are exploring the IVRT method can help us understand the role of electrolyte in stabilization and release mechanism. So another category is in this case is Dr. Robinson liposonic injections. So the challenge for this kind of formulation is that its delayed release mechanism is difficult to measure using traditional IVRT method, and also like a lipid nanoparticle is kind of growing platform for the drug delivers, and we need better IVRT method to understand these formulations. So because of the liposome shares many similarities with lipid nanoparticles, so Dr. Robinson liposonic injection can be really serve as ideal platform to study and facilitate the assessment of future lipid nanoparticle submissions. So here is a summary. So IVRT approach for complex drug products can be challenging and needs more concerted effort to improve. And a new IVRT method was developed that can improve understanding of the in vitro release behavior of complex drug products. And lastly, and we really encourage the development of more innovative and fit for purpose for IVRT methods. So here's the acknowledgement. I really want to thank my always mentor, Dr. Xiaoming Xu, for his guidance, and also all the colleagues and collaborators. So, and here is the challenger questions. So true or false? To develop a suitable IVRT method for complex product, compendial apparatus should be used. And the answer is false. So here is the second challenge question. So which is the following is not critical for developing a suitable IVRT for complex drug products? A, sensitivity, B, selectivity, C, robustness, and D, in vitro in vivo correlations. So, and the answer is D. So thank you for your attention. And this is all finish all the presentation. I will invite all the speakers and panelists to the stage, and we can start the QA section.